Hello, hi, a very happy good evening. I hope all of you are doing good. So this is Krishnaveni and we're talking about the story of life on earth, and that's biology. So welcome to my session. Thank you so much for joining my session well in advance. For all those who are watching my session for the first time, Monday, Wednesday, Friday, I teach a class 12 topic. Tuesday, Thursday, Saturday, I teach a class 11 topic. I hope all your preparations are going on fine. Yes? Okay. So today we are talking about sexual reproduction in flowering plant. This is lecture 4. So today we'll say a goodbye to this chapter. So we'll end this chapter today because we're only supposed to talk about the post-fertilization development, right? So post-fertilization development includes the development of your embryo. Then it also includes the development of endosperm. And we're also going to talk about what is apomixis and polyembryony, okay? Hi Anthony Abhi. Yes, a very happy good evening. I'm good, thank you. How are you doing? Okay, I hope your preparation is going on fine. So today you applied for ICAR, Indian Council of Agricultural Research. Okay, so you are equally interested in agriculture as well. That's super great. Yes sir, a very happy good evening. So thank you for joining our lecture. So please do like, share and subscribe. Okay, so today let's finish off this chapter. So let's do it a bit quickly so that we end this chapter. And next in 12th, we'll be starting with human reproduction. So I will give you a quick recap of human reproduction because that was an equally important chapter for your NEET exam. And after your genetics, your reproduction unit has more weightage. Okay, so shall we start with sexual reproduction in flowering plants? Okay, so welcome to lecture 4 of sexual reproduction in flowering plant. So this is the schedule for tomorrow. So tomorrow once again we are meeting at 5 p.m. and we are talking about the human year. So if you remember, we were dealing with the chapter neural control and coordination. So we were done with the structure of the neuron, the conduction, transmission of impulse in a neuron and between the neurons respectively, the human brain, the human eye. Now, tomorrow we are going to talk about human ear. So, please be present tomorrow at 5 p.m. to finish this chapter, that is neural control and coordination. So, this week I'll finish off human physiology. I will also finish off reproduction unit, after which we will have a mentee. So, your reproduction unit and human physiology unit will be done. So, your revision will be done. So, you will have a mentee. So, probably combining reproduction as well as human physiology because both are equally important. So, please never even think of missing these chapters. So, each of the chapters in human reproduction as well as your human physiology is important, okay? Yes, a very happy good evening, Munna. Yes, I'm good, thank you. How are you doing? Thank you. So, here today you have come well before Shimpi. That's really great. So, you are a sincere student. So, guys, please don't forget to like, share and subscribe. Share the link with your friends. It does not matter if they are NEET 2021 or 2022 aspirants. The more they understand the concept, the more easy it will be for them, okay? So, here we move forward. So, lecture 4 of sexual reproduction in flowering plants. So, what is that we were talking about in this chapter? So, what are the topics that we were revising? So, here we go. So, we were talking about the 4 parts of a flower. So, we spoke about the pre-fertilization structure and events. So, how does the pollen grains develop? So, what is the male part known as the stamens? So, what is microsporangium and how the pollen grains are formed? So, where are the pollen mother cells? So, all that we discussed. So, then we started talking about the pistil, that is the female part. So, megasporangium and the embryo cell. Okay. So, then after the pre-fertilization structure, so now the male and the female gametes are ready. So, they have to meet. So, that process is known as pollination. So, after pollination, it was double fertilization. In between, we also spoke about pollen-pistil interaction, if you remember. So, we also spoke about your artificial hybridization. So, what is emasculation and what is bagging, right? So, then post-fertilization, which we are going to discuss today. So, we are going to see how an endosperm develops. What is the role of an endosperm? What is embryo and how does an embryo develop? So, then seed. So, we are going to talk about how seed development takes place, how seed is important as such. The last one is apomixis and polyembryony. Okay. Yes, Maharani is here. Yes, Shimpi. Hello, a very happy good evening. So, thank you, Shimpi, for joining my lecture. So, please do like, share and subscribe. Ma'am, I like animals and plants both equally. Okay, great. That's really good. Hello Pawan, so I guess you're new to my lecture, so welcome and thank you for joining my lecture. So please do like, share and subscribe. So shall we start? So shall we start from the point of double fertilization so that we can finish off quickly? So I hope the first few parts of the chapter are clear. The first part of the chapter is clear. So we will start from double fertilization and then we will cover the post-fertilization events. Okay. So here I move forward. Okay. So giving you a recap. So first you have 4 whorls in a flower. I will just connect the story so that it is easy. Okay. So you have 4 whorls of a flower. Fine. So what are the 4 whorls? The first one is calyx. So calyx are also known as sepals. Then you have corolla. So corolla is also known as petals. They are the colorful part of the flower. So then number 3. So you have androecium. So androecium is represented by stamens. So we know stamens have 2 parts. One is anther and the other one is filament. Right? The fourth one or the last part is gynoecium. So gynoecium is also known as pistil or carpel. So we are talking about the sexual reproduction in plants. So how does the sexual reproduction happen? Hi Ananya, so thank you for coming here. It's really great seeing you here. So thank you so much Ananya for joining here. So guys, please do like, share and subscribe the session. Okay, so since we are talking about the sexual reproduction, so sexual reproduction involves the fusion of the male and the female gamete, right? So the male gamete here is the pollen grain. The female gamete here is the egg. So the pollen grain matures from the anther and the egg is present inside the embryo sac. So pollen grain can be shed at the 2-cell stage. So what are the 2-cell stage? So you have the vegetative cell and the generative cell. So if it's the 3-cell stage, so here we have vegetative cell and we have 2 male gametes, right? So how is a typical embryo sac? So a typical embryo sac is 7-celled and it's 8 nucleated. Fine? Okay. So what are the different parts of an embryo sac? So you have secondary polar nuclei in the center. So you have egg apparatus. So egg apparatus is towards the micropylar end. So your egg apparatus has 1 egg cell and it has 2 synergid cells. So synergids have what is known as your filiform apparatus. Yes, this is the first part. Okay. So hello, the screen is invisible. Is the screen visible for the others? Yes, the screen is visible for the others. So please refresh your page. The screen will be visible. So then you have the chalacial end. So chalacial end also has 3 cells. It has 3 antipodals. So now here what are our gametes? The egg. So egg and the pollen grains. So these two has to meet. So how are these two going to meet? So this is through a process known as pollination. So for pollination we need pollinators. So how many types of pollinators we have? 2 types. So what are they? First one is biotic and the other one is abiotic. Okay. So biotic includes your animals. Of them the majority are your bees. So abiotic includes your wind. Wind is more common and then you have water. So this is pollination. After pollination, you should have pollen-pistil interaction. So after pollination, so first is the production of gametes. So first is production of gametes. So then you have pollination. So after pollination happens, so you have pollen pistil interaction. So after this, suppose if it's like this, so this is my stigma, style and my ovary, right? So this is my stigma, style and ovary. So this is my ovule, right? So here or my ovule is what develops into my embryo sac. So this is how you should link the story. Suppose if the pollen grain comes and lands here. So I am magnifying this picture. So I have a vegetative cell, I have two male gametes and I have the exine layer. So the exine layer has germ pore regions, right? So if the pollen grain is on the two-cell stage, so if the pollen grain on the stigma, when the pollen grain lands on the stigma, if it's two-cell stage, the generative cell undergoes mitotic division to produce two male gametes. But if the pollen is directly in the three-cell stage, so with the help of this exine layer, with the help of the germ pore region, so if it's directly in the three-cell stage, with the help of germ pore, it germinates to form a pollen tube. So pollen tube forms like this and it reaches the micropyle end of the embryo sac. So this reaches the micropyle end of the embryo sac. So on the micropyle end, you have what is known as synergids. These synergids have filiform apparatus. Okay, so this filiform apparatus is a projection or a way through which the pollen tube reaches the cytoplasm of the synergids. Okay, so you have two male gametes. So one male gamete, it fuses with the one egg cell. So this is normal fusion, right? So or this is true fusion. So this is syngamy and this leads to the formation of zygote, which is diploid. Okay, the other male gamete fuses with the two secondary polar nuclei. So here I have two nuclei. So 2 plus 1, it becomes 3. So it becomes endosperm. It becomes primary endosperm nucleus. Since you have 3 nuclei fusing, this is known as triple fusion. And since overall you are having 2 fusion, one is syngamy, one is triple fusion, this process is known as double fertilization. So this process is known as double fertilization. Done? Hi Swami, yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Okay, clear? Fine. So this is what we have been discussing in the previous lecture. If you see, this is how the pollen tube germinates and this is my micropyle end, this is the chalazial end. So these 3 are antipodal cells. So here I have my filiform apparatus. So I have small projections like this. So the filiform apparatus are here. So then my one male gamete fuses with the egg cell and the other fuses with the endosperm. So other fuses with the secondary polar nuclei. So this becomes the endosperm. This becomes the endosperm nuclei. So this becomes the endosperm cell which consists of the zygote. Fine. So now look here. So after double fertilization, I have inverted the structure. The synergids degenerate after fertilization. The antipodal cells degenerate after fertilization. So this becomes the primary endosperm nucleus. This entire thing becomes the primary endosperm cell. Okay, so here I have my zygote. So this is my entire double fertilization process. Now after fertilization, what is going to happen? That is post-fertilization structure and events. So double fertilization is followed by the events of endosperm and embryo development. So now we have primary endosperm cell. So we are going to talk about how this endosperm is going to develop and how the embryo is going to develop. So we all know zygote develops into embryo, right? So this is how post-fertilization development happens. So literally speaking, ovule develops into seed and ovary develops into fruit. So that is what we expect after a flower. So once a plant's flower, it is supposed to give us a fruit and a seed, right? So that is post-fertilization events. So first let's talk about endosperm. So before the zygote develops into embryo, the endosperm development happens. So that is what is written on the slide. The endosperm development precedes the zygote or the embryo development, meaning it happens before the embryo development happens. Why should the endosperm develop first, or what is the role of endosperm? Why is the law of nature such that the endosperm has to develop first? So let's look into it. So why so? Primary endosperm divides repeatedly. So it has 3N. So the ploidy is 3N. So it is triploid endosperm tissue. So group of cells form a tissues, right? So this primary endosperm cell, each with a ploidy of 3N, it divides repeatedly to form a tissue. That is primary or triploid endosperm tissue. Okay, so the cells of this tissue, they are filled with reserve food material. So reserve is storage. They are filled with food materials that are used for future. Okay, so these food are in turn used to nourish the embryo. For example, if you are going to get admitted into a college, okay, so you ensure you have enough fees amount. So very simple, let's not go all that. So if you are going out, and if you look, the clouds are raining, so you put an umbrella in your bag, right? So that is the simplest precaution you take. So similarly, if an embryo or a zygote is developing, so zygote develops into embryo. So right after fertilization, it is called as zygote. Zygote develops into embryo. Okay? To ensure that the embryo develops properly, someone has to nourish the embryo, right? So that nourishment is done by endosperm. So that is the reason endosperm development takes place first. Okay. So the endosperm cells, they divide further, they form a group of tissues, and these tissues store reserve food material so that they can nourish the growing embryo. Clear? Yes. So the most common type of endosperm development is primary endosperm nucleus. So PEN is primary endosperm nucleus. So this undergoes successive nuclear divisions to form a lot of free nuclei. Free nuclei means it does not have the formation of cell wall, only the nucleus divides, that is karyokinesis happens, cytokinesis does not happen. So this forms free nuclear endosperm. So cell wall formation occurs later and becomes an endosperm cell much later. Okay, so the number of free nuclei formed varies from one plant to the other plant. Fine? Okay, so endosperm, you can see the coconut water in a tender coconut. So this coconut water is very good during summer season, right? So it hydrates your body, it nourishes your body. So it is said that if you have tender coconut water every morning, it's good for the glow of your skin. So of course, coconut itself is completely useful to human, right? So it is known as Kalpa Vriksha because all the parts of the coconut tree is useful to human. So of course the coconut water is also very healthy. So this coconut water in a tender coconut is the free nuclear endosperm. So it is made up of thousands of nuclei. Don't get confused. So we have primary endosperm cell, right? So this undergoes divisions and it forms many cells. So thus this forms a tissue, right? So initially it only divides the nucleus. First it forms karyokinesis, then cytokinesis happens. Okay? So your coconut water in your tender coconut is made up of many types of nuclei. So surrounding the white kernel is your cellular endosperm. So first karyokinesis happens, that is the coconut water. Then your cytokinesis happens, that is the white part of the coconut. So first your nucleus divides to form many nucleus, and certain nucleus are with cell wall. Fine? Okay. So endosperm may be of two types. So the endosperm may be completely consumed by the developing embryo. So the ultimate role of embryo is to nourish the growing embryo. The ultimate role of endosperm is to nourish the growing embryo. Sometimes the growing embryo might completely consume the endosperm. So before actually the seed matures. Okay. So example, peas, groundnut, beans. So all of your leguminous plants. Okay, so all are leguminous here. So you can also say they are also dicot. So in some cases the endosperm persists throughout. So this is castor and coconut. Okay, so these are the two types of endosperm development. Now we are talking about the development of the embryo. So we all know we have a micropyle end, we all have a chalacial end. So in the micropyle end you have the egg. The zygote is towards the micropyle end. So the embryo development takes place towards the micropyle end. So the zygote divides only after the endosperm is developed, and then it forms the embryo. Okay. So embryogeny is the early stage of embryo development. So embryogeny is similar in your monocots and your dicots. So in your first you have your fertilization. So fertilization leads to the formation of zygote. So zygote leads to the formation of pro-embryo, which is globular, heart-shaped, and then it develops into a mature embryo. So these are the stages of development. So dicot embryo, it consists of embryonal axis and two cotyledons. That is why it is called as dicot. It has two cotyledons, right? So the embryonal axis above the level of cotyledon is known as epicotyle. So if I have two cotyledons, so this is my embryonal axis. So here I have my plumule, right? So this part is epicotyle, this part is hypocotyle. So hypocotyle develops into the root, epicotyle develops into the shoot. Yes, endospermous and albuminous are the same. Yes, no doubt. Okay. So root tip is covered by root cap, no doubt. So your monocot embryo, your monocot versus dicot embryo. So in monocot you have what is known as an aileron layer. So aileron layer is the protective covering of the endosperm, and your endosperm and your zygote is separated by scutellum. So you have a cover for your radicle, that is cleorrhiza, and for plumule, that is cleoptile. Okay, fine. Sorry. So moving further, so we will talk about seed. So we are done talking about two things. The first part, so the first part we are done talking about the endosperm development. So then we spoke about the development of the embryo. So basically embryo develops into seed, right? So embryo is nothing but your ovule, right? So ovule develops into seed, and of course the ovary develops into fruit, no doubt. Okay. So now we are going to talk about how the seed develops. So how the seed development happens and why it is there. Is there ploidy of cleoptile? So cleoptile is 2n. Okay. So ploidy of cleoptile is diploid. Alright. So moving further, seed. So angiosperm seed is the final product of sexual recomposition. Fine. Sexual reproduction. So seed is fertilized into ovule. So seed is inside the fruit. So radicle is root, radicle is diploid, then plumule is shoot, that is also diploid. Okay. So only gametes are haploid. So after your fertilization happens, it is diploid. So zygote is diploid. So zygote develops into embryo. So that is also diploid. Okay, Anthony? So seed is fertilized into ovule, and inside seeds are present inside the fruit. So seed consists of seed coat. It has an cotyledon, it has an embryonal axis. So these are the three parts of a seed. So seed has a seed coat, it has cotyledon, it can have one or two cotyledons, and it has an embryonal axis, that is epicotyle and hypocotyle. So cotyledons are simple structures which are swollen due to the storage of reserved food material. Fine. So seed can be non-albuminous or ex-albuminous. So no residual endosperm is left. So when do you say X? When it is over, right? So that is why you call it as X. So when the endosperm is completely consumed by the developing seed, so those kind of seeds are known as non-albuminous or ex-albuminous. Example, pea and groundnut. So albuminous is when a part of endosperm is retained at the time of seed maturation. So this includes wheat, maize, barley, castor. So this basically includes your cereals, okay? So occasionally in some seeds, your nucellus also gets retained. So nucellus is what has your megaspore mother cell, if you can recollect the embryo sac development, okay? So this nucellus also persists in case of black pepper, so pepper and beet. So this is known as perisperm. So when your nucellus is present in the seed development, so such a kind of seed is known as perisperm. Alright. So integuments of the ovule, they harden to form the seed coat. So integument protects the nucellus. So inside the nucellus you have the embryo sac. Okay. So your integuments develop into the protective seed coat. So micropyle remains as a small pore in the seed coat through which the seed helps in the exchange of gas. So facilitates the entry of oxygen and water during seed germination. So seed matures. So as the seed matures, the water content in the seed reduces, okay, and its metabolic activity also slows down if the temperature or if the conditions are unfavorable, okay. The metabolic activity also slows down and it enters into a state of dormancy if the conditions are not favorable. Once the conditions are favorable, it once again germinates. So ovules develop into seed and ovary into fruit. So these two happen simultaneously. Endosperm development and your zygote or the embryo development does not happen simultaneously. First you have the endosperm development, then you have the zygote development, right? But here the development of the seed and the fruit, they happen simultaneously because the seeds are inside the fruit, right? Okay. So the wall of the ovary develops into the fruit wall, that is the pericarp. So you have the wall of the ovary, so that develops into the wall of the fruit, that is pericarp. So you have fleshy fruits like orange, mango, guava. So you also have dry fruits like your groundnut and your mustard. So fruits have evolved mechanism for the dispersal of seed. So this can be through wind, water or your animals. Right? So fruits from the ovary, so other floral parts degenerate and they fall off. So apple, strawberry, cashew, the thalamus. Thalamus is the receptacle on which the four whorls of a flower arises. So that contributes to the fruit formation. So this is false fruit. So you have true fruit. So true fruit is where you have proper fusion. So you have fusion of male and female gamete. Okay, so when I call it as false fruit, so false fruit is when the thalamus contributes to the formation of the embryo. So the formation of fruits without proper fertilization is parthenogenesis. Okay, what is apomixis? Hold on, don't worry, I will teach you. So fruits which develop from true fruits are, which develop from the ovary are true fruits. Okay. So apple, the edible part is the flesh. The edible part is the mesocarp, okay? So few species, the fruits develop without fertilization, that is parthenocarpic. So in false fruit, what happens? Fertilization happens, but thalamus also contributes. But in parthenocarpic, fusion itself does not happen. Clear? Fine. So example, banana. So parthenocarpy happens through the application of hormones. So seed is advantageous to angiosperm. So hi Natasha, yes I'm good, thank you. How are you? So thank you so much for joining my lecture. So please do like, share and subscribe. So seed is advantageous to angiosperm. So just like you need progeny to sustain your population or your community, so for plants seeds are very important. They sustain the seeds to ensure that their community or their population persists. So one, it is the most important output of the sexual reproduction. So it also helps in pollination as well as fertilization, right? So seed formation is dependable on the environment. So seeds have better adaptive strategies for dispersal to new habitats. So they help new species colonize in new area. The seeds also store reserve food material. They nourish the seedling. They also provide protection to the young seedling. So they generate new genetic combination, that is they in turn produce variation, which in turn is the basis of evolution, and they are also the basis of agriculture. So dehydration and dormancy of the mature seed is crucial for the storage of the seed. Suppose if I am going to store lemon seeds. So all of us know Nimbu has seeds. Yes, lemon has seeds. So I am going to store the seeds of the Nimbu or the lemon, right? So I should know what is the dormant period of the seed, and I should also know what is the dehydration. So what is the least amount of water that is present and how I can preserve them is very important. Okay? Fine. So what is viability of seeds? So viability talks about the functionality of the seeds. So certain seeds are functional for a long time. They live for long. For example, if you give me a mango seed, I can keep the mango seed for 1 week with me as such, and then I can plant it. Even then the mango tree will go. So the viability is much longer, right? So for some plants, the seed viability is very short. You have to plant within 2 to 3 days, otherwise the seed loses all its functional abilities. Okay? So there are some plants which are viable for hundreds of years, for thousands of years. So no, you're talking about the viability of pollen grains. I am talking about the viability of seeds. Okay? Yes ma'am, good evening. So thank you for joining my session. So please do like, share and subscribe. Okay? So there are some plants whose seeds are viable for 2000 years, 200 years, for almost 10,000 years. So from fossil, if you take up the seed, so it is able to still grow, right? So in that case, that is viability of seeds. Now, the last topic for the day is apomixis and polyembryony. So there are certain families known as Asteraceae, that is your sunflower family and grasses. So Asteraceae is a family that consists of sunflower, etc. And you have grasses which belongs to monocot. So in these two plants, you have a special type of asexual reproduction. So this asexual reproduction, so it mimics sexual reproduction. So what happens here? So what happens here, or how does this happen? So here the egg cell becomes diploid due to the contribution or due to the folding of nucellus. Here also no direct fusion happens, but because of the fusion of nucellus, it forms an embryo. So this is known as apomixis or polyembryony. So here each ovule has an embryo. So that is the reason it is known as polyembryony. So this is known as apomixis. So in certain condition of apomixis, each ovule can have more than one embryo, that is polyembryony. So polyembryony is seen in your citrus and in your mango. Okay, yes Dharmi, a very good evening. So welcome to my session. I hope you're doing good. So thank you for joining. So please do like, share and subscribe. Is this clear? Apomixis and polyembryony? Yes? Okay. So one last thing I want to tell is the position of the ovule. So your ovule has funicle. So it has micropyle end and it has chalacal end. So in a mature ovary, based on the relative position of all these, you have many types. Okay, let's see what are the 6 different types. So an atropous or orthotropous is the same. So atropous or orthotropous is the same. So here the funicle, the micropyle, the chaliceal end all lie on a same line. So everything lies on the same line, that is orthotropous or atropous. Okay, fine. NCERT में नहीं है, but this is important. Okay, fine. The next one is anatropous. So anatropous. So this is straight line. So anatropous is when it is rotated by 180 degrees. When it is rotated by 180 degrees, and this is the most common type. So here the funicle is near the micropyle end. So if you can look into the picture, this is the funicle, this is the micropyle end. So here when it is rotated by 180 degree. Okay. The next one is amphitropous. So amphitropous is when the body of the ovule is curved. Fine. The next one is hemianthropous. So this is 180, this is hemi of this, half of this. So this is 90 degree. Fine. So camellotropous is when the body of the ovule is at 90 degree only to the funicle. So here the entire thing is at 90 degree. Right, but here it is at 90 degree only to the funicle, not with the micropyle or the chaliceal end. So such thinness is when it is rotated by 360 degree. Okay, so these are the different positions of your ovule with respect, different types of your ovule with respect to the positions of funicle, micropyle and the chaliceal end. Clear everyone? Yes. So this chapter is very important. You will have at least 3 to 4 questions from this chapter. So please prepare well. So today we are done with sexual reproduction in flowering plants. So Wednesday I will be starting with human reproduction. I will try to finish off human reproduction by Friday. So then we might have a quiz. So meanwhile in your class 11, I am talking about human ear tomorrow. So please join me. I will teach you the structure of human ear in short. So after human ear, we will be done with the neural control and coordination. Then I will start chemical coordination and integration. So we will simultaneously finish human physiology as well as human reproduction. So both are 2 important units. So please don't miss them. Okay. So if you have any doubts, so please feel free to contact me. So hi Bhavya, very good evening. So thank you for joining me. So please do like, share and subscribe. So animal kingdom is done. So I have taught animal kingdom almost 3 times. All the 3 times are done. So please refer to the video if you can find. So if you cannot find the video, so please contact me or ping me through Telegram. So I will give you the links. Okay? Yes? So please subscribe the channel so that you get access to all the videos of my lecture. I have taught the entire biology, class 11, class 12, botany, zoology fully once. So please look into my lectures. So please slow down the lectures if you feel I am fast. If you feel I am slow, please increase the speed and watch. It will be a good revision. So I have solved enough questions in each chapter. So please look at them as well. Okay? Yes, thank you so much. So you guys make the lectures very, very interesting. So tomorrow at 5 p.m. we will talk about the human ear. So don't miss the session. So this is me wishing you more and most good luck, good health as well. So stay tuned and bye.